Thank you again for inviting me, and I'm here to sort of talk about our recent R34, as the other members of this section are, and I'm going to talk about cowbirds. So I think I'm the only bird presentation here, and I wanted to do something a little bit different. I wanted to take you back to sort of late 1990s neuroscience, and this is a finding that I did when I was finishing up my postdoc and started in my lab. And we were recording, or I was recording in freely moving zebra finches. And I noticed when the animals were lightly sedated or sleeping, that if I presented a stimulus in the form of the song, typically it was the bird's own song, I got a very strong response in the song control system known as the HVC's nucleus, as well as in the auditory forebrain areas that are just 2 synapses away from the HVC. But what was really striking to me at the time and still now is that when the animals were awake, the responses didn't change that much in the auditory forebrain, but you got a complete suppression, or you could get a complete suppression in these sensory motor areas that control singing. And we followed up on these studies and showed that we could reverse this trend by playing around with neuroadrenergic receptor blockers. And the idea here that you could get such strong modulation of auditory responses in these higher order areas with behavioral state, awake versus aroused or sleeping, really made me think about, is this possible that these types of systems might also modulate sensory processing in social contexts, in different social contexts? And then the big question was, but how do you quantify social context, and what would be a good model system to study that? So I thought hard about this, and I really wanted to look at an animal model that had really interesting, complex social behavior, but that was also a little bit bigger than the zebra finch. So if I wanted to add more hardware on the head without changing the behavior too much, I could still do that. So I decided to move to the brown-headed cowbird. This is a highly socially gregarious bird. Males sing songs, and they sing it in different contexts, male-directed, female-directed. A surprising feature that we discovered in the last couple of years is that the song is incredibly stereotyped, just as stereotyped as the zebra finch, maybe even more. But females also play an important role in these social interactions. They have specific calls. But in addition to calls, you also have these postural displays. Males have different displays when they sing to males or to females. And females actually play an important role in these courtship interactions. In addition to that, you have other postural displays. You have so-called head-up displays, which show aggressive interactions. And then we even have seen some interesting group-level behavior synchronization, where males will switch from all doing male-directed song to female-directed song. So the cowbird really it's suggestive that it has a very complex social system. And so we were interested in doing that. So what we built in collaboration with my colleague Kostas Danielidis over in the computer science department, and then also recruited Vijay, who's a computational neuroscience, as well as Firuz, who's involved in designing electrodes for us, is we built a so-called smart aviary. And this is an aviary that's about a 10-minute walk from my lab. It's equipped with 10 computer vision cameras, an array of microphones, and other devices. And the idea is to follow interactions between every possible bird in this aviary. And so typically for breeding season, we look at about 7 females and 5 males, so 12 birds. And we look over the entire breeding season. And the idea is to collect information about where the birds are and follow their tracking, analyze pose, and then also locate sound, where the birds are when they produce the sound and the types of sounds that they produce, and use all that information to eventually come up with a new variable that in neuroscience we haven't really talked much about, is social context. And so with Vijay, we're trying to use methods from statistical mechanics like Ising models or Potts models to try to collect all our data and establish a model for social context. And in parallel with Firuz, what we're doing is developing a battery-less, so without a battery, but a wirelessly powered and wirelessly transmitting neural recording device. The idea would be to and this is really important for me, I want to be able to record neural activity eventually in birds that look exactly like the other birds. I don't want a big cap on the head that could impact social interactions. And then I want to eventually put all this together to try to ask questions about how does social context, which we can compute or quantify, affect how the brain processes social information. And I think the songbird, and this is my last point, is a really interesting model system because the social signals here are acoustic events. And so they have discrete onsets and discrete offsets, and we can look at how the brain responds to these social signals under different quantifiable social contexts. And I just want to thank you, and I thank you, the NSF previously and NIH now, for believing in this general approach.